Hello, everyone, and welcome to chapter 1 of our lecture videos. And I'm just going to go through a couple things on our Canvas page first. When you open up our Canvas page, you'll notice this link to our Bio 41 YouTube channel where I'll put all our recorded lectures. And then you'll see a link to our spring syllabus. And I'm just going to take you through a little bit kind of what is expected of you in this first week. And I know there's been some questions about the textbook. And, I know this how to register for the MyLab Mastering doesn't work anymore, and I apologize for that. I had used them in the past. So basically what you guys need is you just need any version of the textbook. It can be a paper format, it can be electronic, but any version of the textbook will do. And if you download the syllabus, you'll see the title of that textbook. And again, it can be any version, it can be an electronic, you can buy it through the bookstore, you can buy it through Pearson, you can buy it on Amazon. So feel free to get any version of this textbook, the Campbell Essential Biology, but you will need a textbook. What's nice about getting it through Pearson is Pearson gives you a lot of extra study tools when you go into the Pearson website. If you buy the electronic version through Pearson, you see a lot of extra study tools like practice tests and so forth. So if you do want to buy it through Pearson, you'll have access to a lot of other study tools, but you do not have to. I don't assign any assignments through Pearson. For this first week, you'll notice some key assignments that are due. And the due dates are not loading, but they should say February 11th here. The Who Am I discussion, the syllabus quiz, and then watching these 2 videos and submitting 2 comments and feedback of online learning. There's our link to our Bio 41 channel page. Let me just go ahead and refresh my screen to get this to load a little bit better here as well. And then if we go into Module 1, which takes us through the first 4 weeks of class, you'll notice that I give you all the PowerPoints, and then I give you active reading guides. And this is kind of like your homework for every chapter. And you will have to do this for every chapter. You'll notice that you have to get through chapter 1 this week, and I'll be a little lenient on due dates this first week because I know some of you do not have the textbook yet. But when you click on this active reading guide, I put it in a Word doc format instead of PDF so that you could easily open it up with Word on your computer and type in as you're going, or you can print it out and write in the answers and then upload a picture of it. So it's a really simple kind of like outline of the chapter. And this assures me that you guys are reading the textbook and then kind of going through this outline. And these should hopefully be really helpful in taking you through the key points that I want you to study in this outline. So please take your time in completing those because you will have this active reading guide homework for each chapter. And you can see that I posted all of those active reading guides for the first 7 chapters. We're getting through a lot of material these first 4 weeks. It's about 1 to 2 chapters per week. So make sure you're staying on top of these, completing these active reading guides, reading those chapters in the textbook. And here you'll notice below the active reading guide document, you see where it says the submission. So this right below where I give you that active reading document, you'll submit that chapter document for 5 points there. So not worth a lot of points, but every little point adds up. And hopefully this is a way that you can kind of build up some points doing those homework assignments. Eventually, not this first week because you have enough assignments, but I might throw some weekly quizzes in there as well as weekly discussion posts, but I will hold off on that as you guys are getting used to Canvas. For this first week, focus on these introduction activities, that Who Am I discussion, the syllabus quiz, and posting 2 comments about these online learning video feedbacks, which kind of are great videos just to give you a taste of what it's like to take an online class. And then feel free to work ahead on your active reading guides, which again are those guided notes through each chapter, because again, we have 7 chapters to get through. At the end of each module, we'll have a lecture exam. And again, what you don't see in this first month's module are maybe some more discussion posts and weekly quizzes that I'll upload the week of. And by the end of this module, and a few of you have already started doing this for me, you need to choose a science-related documentary. I'm sorry, it says human health documentary. It's any contemporary science-related documentary, and then you need to just email that documentary to me so I can approve it by the end of this module. And I'm going to go ahead and update this right now. So choose and email to me any science-related documentary. And that kind of takes me through my opening introductions, and then we'll jump right into our first PowerPoint. So you have a lot of information to get through in 7 chapters. And what I try to do in my PowerPoint lectures is keep them short and to the point. You'll notice that the PowerPoints themselves can contain anywhere for up to 100 slides. And I probably won't cover all those slides every single lecture. I'm going to try to focus on information that I want you to know within each chapter and that I'll test you guys on at the end of the lecture. I don't provide you with any study guides, but those guided kind of reading activity outlines that you fill out for every chapter will hopefully serve as kind of a good study guide. If you're filling out those chapter review guides well, you'll most likely see some of those questions on the test, and you should be well prepared. So chapter 1 just gives us a little introduction about learning about life and biology. If you are kind of curious about the world around you, and all of us kind of have that inerrant interest in life or that inborn fascination, then biology is probably for you. And I know some of you are taking this class just for a general requirement, but if you have ever looked around you and you're interested in the natural world or in the species or the human body, then we're going to take you through kind of an introductory course for biology and what that means. So biology is technically the scientific study of life. And we're going to talk a little bit today in this lecture, what does it mean to study something scientifically? So we'll go over the scientific method and how do we tell the difference between science and other ways of trying to make sense of the world. So science is this approach to understanding the natural world that is based on inquiry. So it's based on questions. It's a search for information, the evidence, the explanations, and then answers to those specific questions. Scientists are the people who seek natural causes for natural phenomena, and they focus solely on the study of structures and processes that can be verifiably observed and measured. Biology, like other sciences, begins with exploration, and we'll go over kind of the overview process of science and how we collect data. Data is technically the recorded observations. This is the evidence on which scientific inquiry is based. And as you proceed with your scientific exploration, your curiosity will lead to questions, which is the launching point for the next step in the process of science, which is testing. So scientific exploration starts with making observations where you notice something that's strange or interesting, and then you ask a question about it, and then you're going to seek information. A hypothesis is a proposed explanation for the set of observations, and a valid hypothesis must be testable and falsifiable. Some hypotheses, such as the ones involving conditions that can be easily controlled, lend themselves to experiments or scientific tests, whereas other hypotheses involving aspects of the world that cannot be controlled, like ecological issues, can be tested by making further observations. And the results of an experiment will either support or not support that hypothesis. So here we have the testing a common problem using this process of science or the scientific method. And we're going to go over some of these steps here. We'll go over exploring, testing, forming a hypothesis, making a prediction, running experiments, gathering data, interpreting that data, and then drawing conclusions. If this experiment does not support the hypothesis, we will revise the hypothesis, predict with a new hypothesis, and then experiment again. In fine-tuning the question, scientists rely heavily on scientific literature, which are published contributions of fellow scientists. And scientists often meet to communicate with each other in seminars, meetings, and scientific publications. Before their experimental results can be published in a scientific journal, the research is evaluated several times over, very nitpicky by qualified, impartial, often anonymous experts. And this process, intended to provide quality control of the scientific research, is called peer review. So communication is what scientists do when they share their data, obtain feedback, publish papers, replicate their findings, and build a consensus. And then they produce an outcome where they build knowledge, solve problems, develop new technologies, and then this in turn benefits society. So here's an overview of the process of science where we, explore, make observations, we test, we communicate, we share that data, and then we build knowledge and eventually benefit society with it. Why does peer review improve the reliability of a scientific paper? So all of scientific evidence is peer reviewed by sometimes anonymous sources that are very impartial and unbiased. And peer review assures that the scientific data is being collected correctly, it's being explained correctly, and there's no kind of holes in the scientific process of someone's research. So in order for a scientist to get published, it actually can take years for that to happen. So it's a really complex process, and peer review kind of helps keep scientific papers and data, make sure they're not being falsified at all. A theory is a comprehensive and well-substantiated explanation, much broader in scope than a hypothesis. Theories become widely accepted by scientists only if they are supported by a very large, varied, and growing body of evidence. A theory can be used to explain many observations and to devise many new and testable hypotheses. However, a theory, like any scientific idea, must be refined or even abandoned if new or contradictory evidence is discovered. A fact, however, is a piece of information considered to be objectively true based on all current evidence, and a fact can be verified and is therefore distinct from opinions. Many people associate facts with science, but accumulating facts is not necessarily the primary goal of science. The cornerstones of science are actually the explanations that apply to the greatest variety of phenomena. So when we investigate a hypothesis, we'll run usually tests and experiments that include many factors. A variable is a factor that changes in an experiment. A controlled experiment is one that compares two or more groups that differ only in one variable that the experiment is designed to test. The control group lacks or does not receive the specific factor being tested, and the experimental group has or does receive that specific factor being tested. So let's give an example. Do baby turtles swim? The background is here. After emerging from a 2-month incubation period, turtle hatchlings dig out of the sand and then use moonlight to navigate to the sea. Can the juvenile turtles swim in ocean currents, or do they just passively drift? The method of gathering data. Researchers attached tiny satellite trackers to 24 green sea turtles, each between 1 and 2 years old in the Gulf of Mexico. And the results were that the turtles actually moved faster and along different tracks than drifting buckets, suggesting that they actually travel by swimming. And we can kind of see their data shown here, which is super interesting, of tracking these baby sea turtles to show that they do actually swim when they get to the water. The independent variable was tested for the effect upon the dependent variable. The independent variable, what is being changed, was the type of object being tracked, the sea turtles or the floating buckets. And in this experiment, the dependent variable was the speed of travel. And that little red squiggly line shows the movements of one sea turtle over time. So the independent variable is what is being manipulated by the researchers, in this case, the object under investigation, the turtles or the buckets, and the dependent variable is the response or output. In this case, was the speed of movement. The dependent variable is always affected by your independent or manipulated variable, and well-designed experiments often test just one independent variable at a time. Many medical drug trials include a placebo, which is a medically ineffective treatment that allows the placebo group to serve as a control group. Typically, that placebo group does not know that they're receiving that ineffective substitute, and an experiment in which neither the participant nor the experimenter knows which group is the control group is called a double-blind experiment, and the gold standard for any medical trial is the double-blind placebo-controlled study. So how to recognize blind studies? The type of study, it's not blind if the test subjects know which group is which, and if the researchers know which group is which. A single blind is when only the researchers know which group is receiving the treatment or not. And the double blind, which is the most excellent gold standard for testing medical trials, is when neither the test subjects and neither the researchers know which test subjects or groups are actually receiving the medicine that's being tested. Pseudoscience is any field of study that is falsely presented as having a scientific basis. There are several indicators that you can use to recognize pseudoscience. It could be based on anecdotal evidence, which is an assertion based on a single or a few examples that do not support a generalized conclusion. So we can see kind of features of science versus features of pseudoscience. And I'll let you read through some of this here because, again, my goal in these lectures is to get as many people as I can to watch them as possible. So I try to keep them down to 20 minutes and focus on the things that I want you to focus on. We can evaluate scientific claims depending on peer review, the evaluation of work, and then publishing a study. And recognizing a reliable source, you can kind of take yourself through this checklist. Is the information current? Is the source primary? Are the authors identifiable and well qualified? Does the author lack potential conflicts of interests? Are references cited? And so forth to recognize a reliable source. Let's talk about properties of life. An agreed sea turtle displays these properties of life. The order, the cells, growth and development, regulation, response to the environment, reproduction. Which properties of life apply to a car, for example, and which do not? So properties of life would include these properties as shown here. All right, the Mars rover, for example, Curiosity, has been exploring the surface of the red planet since 2012 and contains several instruments designed to identify substance that provide evidence of past or present life, because even as life exists on Earth, it exists in tremendous diversity, and biologists speculate that extraterrestrial life, if it does exist, could be recognized by those same properties described here. A sample of the diversity of life in a national park in Namibia, as shown there. Major themes in biology that we'll talk about this semester, we kind of have these 5 unifying themes: the relationship of structure to function, information flow, pathways that transform energy and matter, interactions with biological systems, and evolution. And we'll be kind of discussing these major themes in this course. So I'll let you read through kind of these, the correlation between structure and function that can be seen at different levels within biological systems like molecules, cells, tissues, and organs. For example, in human lungs, we'll talk about how the structure of human lungs relates to their function, or the structure and function of a red blood cell and how it relates to how red blood cells carry oxygen. Information flow. So for life's functions to proceed in an orderly manner, information must be received, transmitted, and used. Every cell contains genes, which are the hereditary units of information consisting of specific sequences of DNA passed on from the previous generation. So this is how we flow information through our hereditary genetic pool, through DNA. People with type 1 diabetes often have a mutation or error in one of their different genes, which causes the body's immune cells to attack and destroy their insulin-producing pancreas cells. So we'll talk about information flow and how it can relate to genetic disorders. Pathways that transform energy and matter. We'll talk about the movement, the growth, the reproduction, and cellular activities of life. And that's work, and work requires energy. We'll talk about input from the sun and how the sun transforms energy from one form to another to make life possible. And we'll go over all of this in previous chapters as well. We'll talk a lot about photosynthesis and how really kind of the circle of life uses inflow from the energy and from other producers and levels of the food chain to survive. All right, we're going to kind of keep moving along. We'll talk about interactions within biological systems. So the study of life extends from the microscopic level, the teeny tiny organisms, all the way up to the global level of the entire planet, calling the biosphere. And we'll talk about how we have interactions between the complexity of this organization, between the biosphere all the way down to molecules and atoms. So we have interactions in every step of this life organizational chart. And we'll talk as well about the theory of evolution, of where life is distinguished by its unity and diversity. And we'll go through that and many chapters on evolution as well, talking about Charles Darwin and the Galapagos Islands and so forth. So we will spend that occurring with this. And again, these are the major themes of biology to study the green sea turtle. And these major themes are what we are going to talk about in this course. All right, so I'm going to pause there and I'll see you guys all for chapter 2. Thank you for listening. And if you have any questions,